Hello, I am Benjamin FAZILLEAU, business developer at Naobios, a French CDMO offering process development and GMP services for viral products. I am here today to talk about the role of human challenge studies in vaccine development. The availability of vaccines on the global market is critical. With the COVID-19 pandemic, we have noticed the importance of accelerating vaccine development to shorten the timeline for vaccine approval. As new infectious diseases are emerging, challenge studies will be critical to test rapidly vaccines and treatments, even in the absence of widespread natural infections. Human challenge studies are experimental trials where healthy volunteers are exposed to pathogens to better understand how the body responds to infections. Volunteers can be exposed to viral challenge agents to see if the vaccine prevents infection or reduces the severity of symptoms. This method helps to support the vaccine development, gathering more data in a short period of time. A challenge agent is a specific microorganism that is intentionally introduced into the body of a healthy volunteer during a human challenge study and under controlled conditions to study various aspects of the disease, including infection dynamics, immune response, or the efficacy of vaccines. The manufacturing process of a challenge agent follows strict regulatory guidelines as the pathogens are designed to be used in human trials. The challenge agent must be consistent across batches to ensure that all volunteers are exposed to the same level of pathogen, making the results of the study more robust and reproducible. The general steps for manufacturing challenge agents start with the culture of the pathogen using permissive cell lines as cell substrate. After the culture step, the pathogen is purified to remove unnecessary contaminants. The challenge agent is then used for dose escalation study to determine the correct dose that will be administered to volunteers, so high enough to trigger an immune response, but low enough to avoid serious health risk. Manufacturing the pathogen and ensuring its purity, safety, and consistency is a complex task in direct link with the safety of inoculated patients. The challenge agents must be grown, tested, and prepared under strict laboratory conditions to avoid contaminations. At Naobios, we have state-of-the-art GMP capabilities to support GMP manufacturing of viral challenge agents. Between 2021 and 2024, we were able to manufacture 16 different batches of viral challenge agents for CROs, Biotech, and international consortium. For example, we have manufactured an RSV challenge agent for the European Vaccine Initiative and a COVID-19 challenge agent for the Bill and Melinda Gates Foundation. We have also manufactured several BSL-2 and BSL-3 viral challenge agents for respiratory disease, following as close as possible the GMP requirements, performing risk analysis for any technical gap identified. At Naobios, we have state-of-the-art facility, scientific expertise, and a huge track record to support any viral challenge agent projects. We are also supporting viral vaccine, oncolytic viruses, and exosome projects. For more information, feel free to visit our website and our social networks.